This program is brought to you by Emory University.
Good morning, good afternoon everyone, or morning still I guess. Thanks for coming for our special seminar here. I think most of you know Ken. Ken's here chatting with us today about hopefully joining us. That's our hope and our goal here. And many of you are familiar with his work. He is well respected and well recognized in the field of heart failure and transplantation from both clinical education and research perspective. His medical school training was at Jefferson, and then Mayo for all of the subspecialty training, back to Temple, and then at Penn where he runs the Heart Failure Fellowship. He's the director of clinical research and the director of heart failure and transplant research at Penn. And as I said, has made numerous contributions. We had some interactions with him through the Heart Failure Network, which we're part of, and he's on the steering committee for that. And I think today we're going to learn a lot about the breadth of his research as it goes, from one extreme to the other, from the wet lab to the dry lab and back again, or I guess from the dry lab to the wet lab and back again. And so we're glad to have you here, Ken, and tell us all about what you're interested in. And I think we have the lights right. So.
Okay, good. Well, it's happy to be here and to have a chance to present to you and share. Again, this will be a slightly atypical presentation. It's a little bit of a smorgasbord. It is around this theme of heart tissue research. It's been the sort of common denominator for most of my time as a faculty member, really all my time as a faculty member. Since 2004, I've been exploiting human heart tissue as a byproduct, usually of the transplant program, to empower a variety of different types of research. And my goal would be to share some of those types and to put it in the context of what we're all trying to do, is take things from the bench to the bedside and do things at the bench that are either relevant to humans or find out that they're not. And so the nature of the problem that I'm interested in is heart failure, and I don't think this audience needs a review of how important that is in terms of the population impact and cost to our society. I do want to spend a moment highlighting a slide about current standard visions about translational research and how it goes. And a slide like this gives you the impression that it's so nice and linear, that we have preclinical animal studies or cell culture models, and that they tell us what we need to know, and that then it very quickly translates to human, and we do proof of concept clinical trials and phase 2. And really a slide like this, and then we go to population-based and translation to practice. And I think a slide like this is incredibly naive in some ways, because it makes us look so easy. But this is what's actually referred to as the valley of death. For lots of new therapeutics, and so much is lost between what happens here, so things that might be interesting and compelling and actionable, and actually getting them into translation to humans. It looks so simple here, but it's very hard. And I want to make the case and try to illustrate it, that human tissue research, among many other approaches, has a real role to play in helping bridge the valley of death if we use it creatively and in its versatile forms. I also want to highlight this paper. This is from 1989, as you'll see, so it's close to 30 years ago, where this was the first I can identify of a transgenic mouse model causing a cardiomyopathy. And so we've been studying transgenic mice as a way to bridge that gap. And to my knowledge, there's no therapeutic that has come from a transgenic or knockout mouse, as elegant as they are, that has originated at this level and turned into something I use in my patients with heart failure. It doesn't mean that these things aren't valuable. It's that I think they're necessary but not sufficient. I think we need other tools in our toolbox to help bridge this valley of death. That's my point. Unless something really changes in the next couple of years. But based on 30 years, so far not. So I'm going to, again, it's a smorgasbord. There's going to be a few different paradigms that I'm going to highlight through some of my own work, and most of the paradigms are not just my own work, but other people's I've collaborated with, or other people in other labs that I haven't collaborated with, who have used similar paradigms. But I want to highlight a few things here. Let me go back. I apologize. So the first paradigm is sometimes we get curious. We see things at the bedside that strike us as interesting, and we want to study them more. and see, what went on there, what's the mechanisms behind it, is it really real, or when you study it in a more reductionist, controllable circumstance, you find that it's not all it was cracked up to be. It was something, more phenomenological but less interesting. And so one of my first things as a faculty member was that sort of question, right? That we had people, this was the dawn of the LVAD era, or pretty close to the dawn of the left ventricular assist device era, and we were seeing that people on first generation left ventricular assist devices seemed to have reverse remodeling. And we wanted to know, is this real, or is this just decompression of the heart and unloading of the heart? Is there real changes of meaning happening? And of course, we all know how important load is in governing in vivo heart function, right? If you really vasodilate somebody, or you get their preload just right, or the combination of the two, you can really make a big difference in someone's ejection fraction, other cardiac function. And so it's hard to know whether an apparent increase in ejection fraction when someone was on a first generation LVAD really represented intrinsic improvement in cardiac function or not. And we needed a different approach. So that was really our question. So again, this is a picture of first generation LVADs, not because it's what we're using now, but because this is what inspired this question back in the day. This we kind of realized that because VADs induce immediate and sustained and fairly profound left ventricular unloading and also gradual neurohormonal deactivation, this was prior art at the time we were doing these studies, that we knew this would be the mother of all reverse remodeling strategies, because these are potent drivers of progression of hypertrophy and failure, right? So if we are ameliorating them, this is going to be our chance to see whether even chronic end stage, stage D hearts, this was even before we were calling them stage D, could actually reverse remodel. Okay, and the challenge, of course, is how do you assess function during profound unloading? Because as I mentioned, load is a big factor in ejection fraction, et cetera. So there's also, so the approach we wanted to take was, let's take tissues and cells from these explanted hearts, because in those days, it was before the rematch trial, and we weren't using them as bridge to, as destination therapy. Almost everybody was a bridge to transplant VAD. That's how all the new devices still get tested. And so that was a great opportunity, because of course we have a chance to have paired data samples before and after this mother of all unloading interventions. And we wanted to do isolated myocyte studies. This was my K award studies to explore this phenomena. Now, there's challenges to doing isolated myocyte work in human hearts. One of them is that the heart's high metabolic activity means that even 10 minutes of warm ischemia messes up the whole experiment, right? That you get significant degradation of cardiac proteins and messenger RNA, and certainly of contractility, and so you have to take good care of the hearts. Endomyocardial biopsies, which might be a much more convenient way to do this, yield tiny little pieces of tissue, and they're sort of crunched a little bit, and it's not necessarily good for getting isolated myocytes that will beat. Heart transplants are usually done at night, and that's annoying. Due to donor shortages, most normal hearts are used for transplantation, so at the end of the day, sooner or later, you're going to need controls to compare with and without VAD as a benchmark. But we developed techniques. So this slide I'll show at least one other time during this. And we developed approaches that included in situ cold cardioplegia. This is sort of a little commercial for how important it is to play well in the sandbox. We were going to the heart with our buckets, going to the OR with our buckets in the middle of the night to get the explanted hearts, and we were bringing them back to the lab and getting nothing but balled up myocytes week after week, month after month, until one of our surgeons said, maybe I ought to give cardioplegia. I mean, if we were trying to take good care of a heart, that's what we would do. And of course, all of that, as soon as we did that, we started having meager success, and then we had something to work on to optimize. So we learned how to take care of hearts, do a perfusion-based digestion, get isolated myocytes, and do functional studies. We would always put some things in the freezer. We did some muscle strip mechanics as shown here. I'll highlight two data slides because this is very old work. These were both published in Circ. This one is about contractility. So these are representative, they really are representative cell shortening transients, right? So this is cell length on the y-axis and time on the x-axis. A normal cardiac myocyte from a non-failing donor looks like this. It's nice and spiky, and it's rapid shortening, rapid relengthening. A failing one looks much slower, lower extent of shortening and a slower rate of shortening and relengthening. And with heart failure with LVAD support, and again, the average data is shown here, we see improvements in the fractional shortening, improvements in the time to peak, that gets shorter again, and time to 50. And what's shown here is that this doesn't look like normal. It looks better than failing, and it doesn't look like normal. And that's going to be pretty much the blanket thing for the whole LVAD recovery world. It migrates towards normal in most of the signature abnormalities, and it doesn't overshoot or get past it. One other data slide, and then I'll just summarize this part. Is it really reverse remodeling? Okay, the cells work better, but do we have regression of pathologic hypertrophy? Do we have atrophy was a big question. Maybe the hearts are overshooting, and then we didn't really find that. So cell volume and the non-failing heart disease were done actually with a Coulter counter. If you have the right aperture size, you can put isolated myocyte preparations through a Coulter counter. They found that their average volume was almost double in the failing versus non-failing, and with LVAD support, it migrated towards normal, but not to normal and not beyond normal, so they really weren't having cellular atrophy. All the major dimensions were doing the same thing. Cell length was getting longer and then shorter again, but not overly short. Number of binucleated cells, this is kind of interesting, right? That in failing hearts, they actually have twice as many binucleated cells, so this suggests it's trying to do cell division, but not quite closing the deal with cytokinesis. But that doesn't change either. So these smaller cells aren't because the cells are finishing their cell division. And so these two papers made a big splash at the time and suggested that really there was LVAD-associated reverse remodeling at the cellular and the tissue level, that there was improvements in function. It wasn't just an in vivo artifact of an unloaded heart. And it promoted a lot of additional work by me and my lab and my collaborators and other labs. And I'm highlighting some of our studies that basically showed many of the signature abnormalities that in vitro tissue-based assessments permit you to assess improved contractility and calcium homeostasis, the decreased cell size, action potential shortening, improved force frequency, and beta-adrenergic responses, right? We know that our patients with heart failure don't just have abnormal function at rest, but they have impaired contractile reserve, reduced myocardial cytokines. So we had a variety of studies. There's probably, dozens and dozens of LVAD recovery or LVAD reverse remodeling at all these different levels, and we've contributed to that. The interesting part is that now it energized the field of, well, maybe we can use these devices to get some of our stage D patients to actually recover. So we're right now part of something called the Restage HF trial. It's initiated by Emma Burks, who had a big New England Journal paper a number of years ago, but now we're trying to do it in a multi-center case series. 40 patients with severe heart failure due to dilated non-ischemic cardiomyopathy. As you might imagine, if you've infarcted 60% of your heart, you're not going to be a very good candidate for overall heart recovery, even though the isolated myocytes from those hearts often do look better after LVAD support. This is with the HeartMate 2 device, as it's funded by Thoratec. They were recruited at 6 centers, all had EFs less than 25%, all had legitimate indications for LVAD implantation, either as BTT or destination therapy. Pump speed was maximized. It included a protocolized pump speed optimization based on optimal unloading with echo guidance. It also treated aggressively with medical therapy as high or higher doses that we usually use for preventing progression of heart failure in our patients. And LV function was assessed in a protocol-driven echocardiogram-based series of assessments, both before implant, before explant, and after explant. And so I'll highlight, I apologize, some of the interim data here. Again, this study's in progress. It's still in follow-up. So basically, we've enrolled 36 patients, and already 17 have been explanted, and of these, 2 have died, and the others are still, 1 needed transplant, 14 have stayed up, so 14 out of 36. One of the deaths was from suicide, not from heart failure. One much later, and again, others are still ongoing, have not been explanted yet, and are in follow-up. The ones who were explanted data, the data looks nice, 55% average ejection fraction, nice dimensions, nice wedge pressure with the pump down to 6,000, which basically is no net forward flow from the HeartMate 2, and duration of support typically was a year of support before these results were achieved. And so in selected patients with chronic non-ischemic cardiomyopathy, aggressive pharmacologic therapy, optimal LVAD unloading, and regular testing to see whether you're really, some of the studies that have reported really low rates of cardiac recovery with LVAD support either picked the wrong patients or they weren't really looking for recovery. They just said, how many times do we happen to see it? And this is a study that was really designed to pick the right patients, to try hard to make it happen, and to look to see whether it was happening, and then to close the deal with Explant if you had criteria met. And so this was reproducible with explants occurring in 5 out of the 6 centers. And right now, the high rate of explant and remission from stage D heart failure was 44% and 47% overall explanted. So these are pretty high levels, and it looks encouraging, again, in the right patients with the right things. So we don't typically see these kind of rates unless you do it in a concerted way, but I'm encouraged by this. And again, it's not taking people who have had 60% of their hearts infarcted. So again, in each segment of this, I'm going to place acknowledgements. And so I'll add tissue adaptations for when I was at Temple mostly, and continuing a little bit at Penn. And the rematch heart failure trial, these are some of the other investigators, and this was supported by an NIH grant and by Thoratec in the case of the rematch heart failure trial. So I want to go to a different human heart tissue paradigm. That's one that shows that when you really dig in and study heart recovery, you can see evidence of it, and then that can inspire translational clinical research that may actually have an alternative for some subset of patients with stage D heart failure. Now, this is one that we've used, this paradigm, the most, and I'll highlight some of that. Validate a therapeutic target implicated in animal model of heart disease. As I mentioned, there's 30 years of transgenic mouse studies and knockout studies, and I think they're great. And what we need is to find out, do they have applicability to human? And we need a tool to do that. And I think human heart tissue is one of those tools. And so a number of people over the year have come to us and saying, you have a nice biobank, can we use it to explore whether there's relevance, or can we do some parallel studies? So in this case, we did some parallel studies with a young investigator at Penn, Ben Prosser, and we're looking in very new studies at microtubule-associated modulation of contractility. So let me show this to you. So this is inspired by, we know that when we do isolated muscle strips from human hearts, everybody has an upsloping relationship between their sarcomere length and their developed force. The basic length-tension relationship that's the basis for the Frank-Starling mechanism is intact. We don't see any descending limbs here, and whether it's from LVAD-supported, non-failing, ischemic, non-ischemic, they're all upsloping, although the slopes may vary a little bit. And there is much more disease-associated changes in the diastolic function, right? They tend to be stiffer hearts. The ability to, when you stretch sarcomere length, to get better contractility is pretty consistent. And yet, there's also old data like this in the literature. This is from Lynn Warner-Stevenson's group, where they show that when people come in with decompensated heart failure, we'll call this the baseline, and then they get decongested, their stroke volume actually goes up a little bit. And then when they are in sustained therapy, it goes up further. Now, sustained therapy includes some vasodilator therapy. Maybe some of this does too. So it could be multiple factors at work here, multiple changes in loading condition, but this is sort of the opposite of what I just presented. Usually when you decrease filling pressures, you don't get an increase in contractility, an increase in stroke volume. And although it's not a pure experiment, we wondered, is there any basis for this? We certainly do see some patients who come in like this, that they're overly congested, we unload them and their cardiac output goes up, and that doesn't fit with what I just presented here. So what's happening there? It could be changes in their functional mitral regurgitation. It could be changes in their catecholamines, but maybe there's other things at work. And so in that context, this is a really elegant set of studies, again, by this collaborator, Ben Prosser, in Science from a way back, where he takes isolated cardiac myocytes, in this case from a rat. He uses this myotac to put these two manipulators on the ends of the cell and stretch and do a little length tension relationship between contractile force, shown here, as he does 2% increments in length, as shown here. And what you see is the diastolic tension on this individual myocyte mechanics goes up slightly, but the developed force goes up quite a lot. So this, again, is the Frank-Starling mechanism, the active force versus length relationship. And when he does this, he also looked at intracellular reactive oxygen species generation with this DCF fluorescence signal, and found that there's an increase with 8% stretch that's really not nearly as much with the lesser degrees of stretch. In this DCF fluorescence signal indicating reactive oxygen species generation. And they found that either an inhibitor of NADPH oxidase 2, DPI, was able to ameliorate that, or interestingly, colchicine. Microtubule disruption was able to disconnect. So somehow the mechanosensing of this increase in stretch involved microtubules, and that mechanosensing leading to reactive oxygen species generation was very much dependent on microtubules. So stretch, microtubule sensing, NOx2 signaling, and then reactive oxygen species generation. So this got Ben interested in this whole area of what do microtubules do in stretch sensing, and how do they do that? And so he then, when he moved to Penn, did a series of studies with a very advanced new scope, an ARIScan scope that has very high spatial and temporal resolution, with combined with proteins that can decorate microtubules, and started imaging microtubules in vitro. So this is standard confocal imaging of microtubules, not so beautiful. This is with this new scope, the ARIScan one, but as I mentioned, it also has high spatial temporal resolution, so you can do actual videos and track the microtubule buckling during individual contractions. And so that's this EMTB is the one that decorates microtubules, and you can see that the microtubules are acting as a lattice structure within the myocardium, within basically the cytoskeleton of individual myocytes. So this is not the extracellular matrix, this is the intracellular matrix, okay, the cytoskeleton. And when you slow this down a little bit, next video, what you see is this buckling dynamics happening with each and every shortening. And when you have this kind of spatial and temporal resolution, you can actually do more careful analyses of the wavelength of these buckles and their periodicity and their magnitude. And so that's shown in this next slide here. He started quantitatively assessing the buckle occurrence and the buckle amplitude, and finding that it could be modulated by things that were already known in other fields, specifically oncology, to affect the tyrosination and detyrosination of microtubules. So microtubules have post-translation, you can obliterate them altogether with colchicine, or you can manipulate their biology with things that affect the post-translational modification or the balance between tyrosination and detyrosination. To make a long story short, the detyrosinated form tends to result in more attachments to the Z-lines and make the overall buckles smaller in periodicities, shorter wavelengths, and greater in stiffness. The detyrosination can be lessened with two different manipulations. One is with a gene therapy approach, the TTL, so the enzyme that's in dynamic equilibrium phase that is responsible. So there, for the tyrosination balance can be knocked out and favor more tyrosination, which is the less stiff form with TTL. Or you can give a cancer chemotherapy drug called Parthenolide, which also retyrosinates microtubules. And so the buccal amplitude and occurrence tends to decrease with either of these approaches. Either the gene therapy approach with TTL, that does it most profoundly, or to a lesser extent with the Parthenolide. Now, the sarcomere shortening also gets longer, all right? So you have greater degree of sarcomere shortening when you give TTL here. And what's shown here is the peak calcium transient isn't changing. So this is a calcium-independent change, basically based on the cytoskeletal dynamics, that is very encouraging. It's a positive inotropic approach, if you will. So then, that's when he came to me. Finally, we got to why is this illustrating any paradigm? What's happening in human hearts? So we wanted to see in different groups of human hearts that we had banked in our freezer, donor hearts, hypertrophy, dilated cardiomyopathy, hypertrophic cardiomyopathy. And what we found was that detyrosinated tubulin, alpha tubulin, was relatively low in the compensated hypertrophy from non-failing donors, and really truly nearly normal donors. Whereas the tyrosinated tubulin was higher. In fact, it was even a little higher in the hypertrophy, and in the dilated cardiomyopathy and hypertrophic cardiomyopathy, it was definitely a switch towards more detyrosinated, the stiffer form of tubulin. The modulating enzyme doesn't seem to be different in its expression. And so then when you summarize the data from these different groups, healthy donors, compensated hypertrophy, this was set as the arbitrary 1-to-1 ratio, although not necessarily. So this is healthy donors as our reference. Compensated hypertrophy actually had more tyrossinated tubulin and less detyrossinated tubulin. That would favor less stiffness. But then once you get to the pathologic groups, whether it's dilated cardiomyopathy, ischemic cardiomyopathy, post-VAD, or hypertrophic cardiomyopathy, we see big increases in the ratio of detyrossinated to tyrossinated. And this would be expected to favor greater myocardial stiffness, maybe resistance to contraction. When he looked with it, so hypertrophic cardiomyopathy patients going to transplant are kind of interesting, right? Because these patients sometimes get transplanted because their EF has begun to decrease. They have developed systolic dysfunction, burnt out hypertrophic cardiomyopathy. But some of them actually get transplanted because they have bad diastolic abnormalities. And we started looking at the relationship between detyrossinated tubulin, the one that's associated with greater stiffness and an impairment of contractility, and ejection fraction. We found a pretty strong correlation just in the hypertrophs we had in our biobank. And so this was sort of interesting. And most recently, this is data from last week. These are 2 heart transplants we did last week. That same approach where he has the ability to assess viscous effects. This is quite a lot of cells, okay? So this is control-treated cells, vehicle-treated cells, N equal 30, colchicine and parthenolide in 2 different dilated cardiomyopathy cases from last week. So we see that this is the baseline, the DMSO vehicle-treated. This is a shortening profile. It's a lot like those heart failure shortening profiles I showed you in the LVAD story. Both of these black lines are like that, but if you give either colchicine for acute microtubule disruption or parthenolide, you see a pretty nice increase in contractility in these 2. And that when you do that cyclic stretch at different frequencies, you also get a change in the viscoelastic modulus of isolated myocytes that separates out more at higher frequencies, which is what makes it viscoelastic. And so this is sort of an interesting story. So this does look like it has relevance to human, which was the whole point of this segment of the talk, right? Now there have been a number of areas like this. So acute increases in cardiac myocyte stretch stimulate microtubule-dependent oxidative stress signals. That post-translational modification of alpha-tubulin that tends to favor greater detyrossination will tend to make isolated myocytes stiffer. Via microtubule network anchoring to more points, usually Desmond anchoring, and that'll tend to provide a resistive viscoelastic force that limits the amount of shortening. And we realize that when we inhibit that by either a drug that blocks the amount of detyrossination or favors greater tyrossination, we get better contractility, or with a gene therapy approach. And then parthenolide has significant positive inotropic effects in failing human cardiac myocytes as of last week. And that would likely be an energetically favor, right? Where we have a dampening force of contractility, taking it away without affecting the calcium transient. I didn't show that, but we did calcium transients in the studies last week, and we did not see any change in them. So it really does look like this is from reducing the damping resistance within the cytoskeleton. And so again, the people who had a lot to do with this are Ben Prosser and people in his lab, a few people in my lab as well. This is supported by funding as shown there. And this whole idea of using human heart tissue to extend something really interesting and cool that's been shown in rodent models. I've been a big part of that in a lot of ways, in a very fortuitous way. I'm one of those people who has a big biobank and likes to share it, and in sharing it, I've been a part of very nice studies from a lot of different investigators that get published in very nice journals over recent years, and this is just the past 7. And I think we've advanced science. I think we've helped. This is another way that we cross that valley of death. We find things that look interesting in rodent models, and we find out whether they have relevance or not to human. And a couple of these stories didn't, but most of them have. Another general paradigm is employ advances in human cell and tissue engineering for mechanistic insight. Now, maybe this is cheating a little bit, right? Where what I have in mind here is to present some tissue engineering work that we've begun doing in recent years and are beginning to get some payoff from, taking advantages of the fact that we now can make cardiac myocytes from patients' induced pluripotent stem cells that we can differentiate them into cardiac myocytes. We can get them reasonably mature in 3D models and use those to answer some questions. One of the questions that we address first, because it's probably easier than some of the others, we were talking a little bit about maturation and proliferation studies. Those might be a little bit harder, but we could move on to those soon, is toxicity questions, right? So we know that there's lots of different new chemotherapies, tyrosine kinase inhibitors in particular, that have lots of potential for killing cancer, but also have some cardiotoxic effects. And one of the questions relates to Sunitinib, and that's highlighted here. So TKIs are used widely in the treatment of many cancers, have led to significant survival gains, but a lot of these agents have cardiotoxicity risks. Sunitinib is an interesting one because it's widely used in solid organ tumors, especially renal cell carcinoma, but others too. It has a high level of causing hypertension. About 47% of patients get hypertension. A smaller percentage could get left ventricular dysfunction, but it's not trivial. It's 10 to 21% in the cohorts that have studied this, including one at Penn. That nearly everyone who gets cardiotoxicity also gets hypertension at first. And so the question arises, is the cardiotoxicity because the hypertension is unmasking it, or is the hypertension, is the increased loading conditions actually contributing to the cardiotoxic risk, right? You can imagine a myopathic heart or emerging myopathy would show up more if there's hypertension, but it could be that it's actually potentiating this. And this is not an easy question to address in vivo without a large, carefully protocolized clinical trial. And the oncologists don't actually like treating hypertension really aggressively in these patients. And so we decided to use the tissue engineering approach to study this. But in addition, there's a lot that's not known about, or mechanisms and the relative contraindications of these different signaling pathways, especially AMP kinase signaling, to the sunitinib-induced cardiotoxicity, and particularly the effect of loading conditions and the hypertension really was unknown. And so we've begun studying this. So our hypothesis was that increased in vitro afterload actually lowers the threshold for sunitinib cardiotoxicity. The approach we took was an engineered heart tissue model construct that mimics a syncytial three-dimensional arrangement of myocardium, including biomechanical loading conditions and the ability to manipulate them. We wanted to establish an in vitro metric of cardiotoxicity that we could track in a variety of different circumstances and define the dose dependence of sunitinib and see if in vitro it has anything to do with the plasma concentrations that people have in vivo, because if it doesn't, then it's not really a relevant model. Compare toxicity at alternative afterloads is the money question we had, and of course we also wanted to begin transitioning this from the neonatal rat cardiac myocytes that many of us use at the beginning to, in these engineered tissue models, into some of the iPS-derived human cardiac myocytes.
Could I just ask you a quick question?
Yeah.
What do you mean by cardiotoxicity? What are you seeing?
I'll show you.
Okay.
I think the data slides will show that. So let me delay it three slides while we introduce the model. So this is a model system we inherited from Chris Chen, who's now at BU but was at Penn until a few years ago. It basically takes a silicon mold and that is microfabricated and that can be filled with a combination of cells, media, collagen, and serum and growth factors. That over time, not much time, like a day, can self-assemble into microtissues that are each about 500 microns in length, so half a millimeter in length. These are not patches that we're going to transplant onto people's hearts. These are in vitro model systems to study biology. This is a fibroblast-only video that shows how the self-assembly process, the same self-assembly also occurs with cardiac myocytes, except at the end they beat. So shown here, this is a bird's eye view up here, and then a side view here. We see that over the course of 24 hours, the tissues are forming, right? They're pulling away from the outer walls of this little pool. They're getting hung up on the knobs of these cantilevers that are part of the microfabricated mold. And they're generating increasing tension that you can see the way they're bending in these cantilevers at the top. And that then plateaus off. So they self-assemble into a microtissue, even if you just put fibroblasts and the other collagen and media in the well. But you can also put neonatal rat cardiac myocytes in, and I think this video is not going to run, but maybe it will. It didn't earlier. So the only difference here is that it shows that when you have this isolated muscle strip. Now it's comprised of neonatal rat cardiac myocytes shown here. You're going to get both the static tension that you saw with fibroblasts only, but then you'll get intermittent increases in tension when the syncytium of isolated myocytes starts to contract. I'll show that on a different one. Important thing is that these are quantitative, right? Because you know the spring constant, or you can calibrate and measure the spring constant of these cantilevers, and so they're not just posts to keep the cells from balling up into a mass in the middle. They're actually gauges of contractility. And their deflection, both under their static deflection or diastolic or their active shortening deflections, give us a real-time measurement of contractility. And so you can see that in these videos that I think will work today. Now this is again a proof of concept with human embryonic stem cell-derived cardiac myocytes. The same works with iPS cells that I'll show later. You can then, when they're made of cardiac myocytes, of course, they can begin to beat and pull in the cantilevers, and the deflection of the cantilevers can tell you something about the contractility that's being generated. You can do things like force frequency responses, and you get faster deflections and motions inward. I also want to mention one important wart about the current state of the art for most of these models. So we see that we can pace them at higher frequencies, as shown here, but we also see that the diastolic tension's going up quite a lot, and the developed tension's actually going down. So this is a negative force frequency response, the kind you would observe in pathology, or right, because these are human now, they should have a positive force frequency response, or that you would observe in immaturity. And in this case, I strongly suspect it's because of immaturity. We weren't trying to make a pathologic model, but these are done from stem cell-derived cardiac myocytes that are still in a fairly early phase of development, right? We're studying them at day 20 to 27 after the differentiation protocol begun. Yeah.
How many cardiomyocytes you?
So if you can ask the question, you got to use the microphone because people are listening remotely.
So the question was, how many cardiac myocytes are in each tissue?
Yes.
So about 500. So we will see them. Go ahead.
And the contraction in the middle is lesser, or do you know, like of the cells?
Hang on a second. Wait till you get
on the
floor. Hang on.
The contraction of cardiomyocytes is.
Hang on. Push the button. Yeah, turn it on. We got people at tech listening.
All right. Okay. The contraction of the cardiomyocytes in the middle is changing compared to when they're moving towards the poles.
In general, they're well distributed throughout.
Mm-hmm.
And also in general, they're not densely distributed throughout. So one of the things that I'm unhappy about, and this is for the people at Tech who I want to collaborate with, is that this model is a proof of concept itself that you can apply these different approaches, cell engineering, tissue engineering, to get a syncytium of myocardium, a fabricated microtissue. But and there's many different labs around the country and the world that are working on these models, but none of them are perfect in a variety of different ways, and I'll have a chance to discuss that more. But suffice to say, their cells are distributed throughout the tissue that self-assembles. They're not as dense as mature myocardium. The cells that comprise it are not as mature myocardium. And so this is an evolving model of human myocardium that might be used for certain purposes, but if we really want to model disease, we're going to have to make a better model that's more mature and that we can start creating some pathologic inputs. For toxicity studies, as I'll present here, they're probably the low-lying fruit of applications, and so I'll have something to show. So for an example, you asked earlier what do we define as toxicity? So one is caspase activation. That's going to turn out to be our best marker. If we look at Sunitinib versus vehicle, this was, I think, 10 micromolar of Sunitinib versus staurosporin. Staurosporin is a big-time caspase activator. Sunitinib, pretty significant as well. We see the treatment duration. Most of this action happens in the first 8 hours, and it's already beginning to wane with longer periods of time. We found that there's a nice dose response to Sunitinib, to barely threshold doses of 0.2 micromolar and to what I think is excessive 10 micromolar here, but that's our most robust responses. But with 1 micromolar, which is definitely clinically relevant, that was one of our questions, do 1 micromolar concentrations that are observed in vivo in patients getting Sunitinib cardiotoxicity, yes, that does look like we see a significant signal. We also look at things like trypan blue exclusion, just to see how many cells are really dying later on, a later time point, more like 24 hours, and we see that this early caspase activation isn't being, it is leading to ultimate cell death and the same kind of relationship between here and here. So, but caspase activation is our best metric, and that's what I'm going to present mostly. We also see that at the same time this is happening in terms of caspase activation and cell death based on trypan blue, we also see that there's a dose-dependent decrease in spontaneous beating frequency. We can still pace these cells, but their spontaneous beating tends to go from down over time. We see that, importantly, this is static tension in the pale purple bars and dynamic active tension. Active tension is going down as we're doing a dose response to sunitinib, but even the lower dose has a significant decrement. And we also see the same with diastolic tension going down over time. We see the excitation threshold isn't really changing. So this is how much voltage does it take to make the cell beat in a pacing mode. That doesn't really, or the tissues beat. And we don't really see any change in the maximum capture rate as these cells are suffering under sunitinib toxicity tissues. We looked at a little bit of mitochondrial function data, and that's shown here. Sunitinib using TMRM as a measure of mitochondrial membrane function. We started seeing that with CCCP, which is a positive control. We see marked decrease with vehicle control. It's up here. Sunitinib caused a mild decrease, and it's hard to know whether this mild decrease is just some cells dropping out. This required actually dissociating cells from intact microtissues. It's a technically demanding experiment, but it shows it's possible to do flow cytometry on those cells that come from a series of microtissue experiments. We see relative ATP levels drop as well. Again, I don't know if these decreases are just proportional to the cell death or they're things that precede the cell death. We really don't know that yet. We did look at an AMP kinase inhibitor because there was some suggestion in the literature that that might make a difference. Activator, I apologize. And that did not ameliorate the sunitinib toxicity. So that hypothesis was not supported in this model. Sunitinib cardiac tissue, in microtissues, now we want to get to the money question, right? Does it affect does increased cantilever stiffness. So you can control, at least at the beginning of the experiment, the stiffness of the posts onto which these things are mounted. And so we wanted to see, would that make a difference in how much cardiotoxicity? This is experiments in still neonatal rat cardiac myocytes. So with sunitinib on soft pillars, we see this kind of magnitude of increase. And again, the fold change is shown here on soft pillars. But with the stiffer pillars, try the colors reverse here. We see a greater increase from the vehicle to stiffer pillars in neonatal rats. And so that's shown here. In stiff pillars, the fold change of caspase activation is quite a bit greater than on soft pillars. So that begins to suggest that maybe that increased afterload or hypertension we see in vivo isn't just unmasking toxicity, it's actually changing the threshold for toxicity, as suggested here. And finally, we started doing these most important experiments in human-derived cardiac microtissues. And again, with the 1 micromolar sunitinib versus 10, we actually see a greater dose response. So the human microtissues, if anything, are more sensitive to sunitinib than to higher dose of sunitinib than the rat. And more importantly, when we compare stiff pillars versus soft pillars, the change between human is even greater than the change in rat. So it does look like increased afterload actually lowers the threshold for sunitinib toxicity based on the metric of caspase activation, but in some early data, there's not enough numbers to analyze, also in terms of contractility. So in engineered heart tissues, there's dose-dependent and time-dependent cardiotoxicity with clinically relevant concentrations of sunitinib. The caspase activation is the most reliable signal we have and parallels other metrics of cell death and defects in active force generation and mitochondrial function. The AMP kinase activator, ACAR, does not mitigate the in vitro sunitinib cardiotoxicity. This would benefit from a few more experiments with some other rescue strategies. And increased afterload clearly enhances in vitro sunitinib toxicity and provides a rationale for more aggressive antihypertensive therapy in sunitinib-treated patients. I think this is beginning to fit with some literature, and it's forming the basis for some trials we're thinking about doing in the Heart Failure Network. I think, Andy, you'll hear about this at the upcoming meeting on Thursday, where we're going to be proposing, again, some disease-modifying heart failure therapies, including the vasodilators we like to use, or beta blockers, to try to protect people at risk for these situations. So again, the people who have helped with this are my graduate student Rachel Truitt and other postdocs in the lab, people from Chris Chen's lab, and the funding shown below. I have one other paradigm here that I guess I'd like to present, but I want to leave some time for questions, and it might be a little more important to skip ahead. I'm going to skip ahead a little bit. So I think I've illustrated the point here that one of the attributes of human heart tissue research is, by definition, it's always clinically relevant. We're getting tissues from our patients. It's clinically relevant, whether it has certain warts that make it need a complementary studies in animal models. The one I skipped was one in which we took a human phenomenology and we developed an animal model to study it more carefully, because we do that too. Sometimes we have to go back to the lab and say, okay, it was an interesting phenomena, but what did it really mean? And we have to get more rigorous. And I believe in animal models for that purpose, for sure. But I'm much more excited about using them for something that began with a human tissue observation. I think it also, one of the things that I think we give short shrift to that may be part of the valley of death is, human tissue studies are heterogeneous. Not, what you get out of patients with all their different histories and etiologies and treatments and durations of disease is going to create heterogeneity. And the signals that will show up amidst that heterogeneity are usually more robust. And if you do see things show, so it might hurt your ability to publish certain things because there's more variability, but a signal that shows up above that is a robust signal. And I think sometimes our animal models are too homogeneous. It exploits and refines insights provided by applications of clinical therapeutics, and I think the LVAD example is good with that. It allows functional comparisons that are not confounded by in vivo biology. It's one of the reasons we often do reductionist models. In the case of the Sunitinib one, it's very hard to get at that hypertension without a big trial, but now we've inspired a bigger trial, and I think that's valuable to have a controllable situation with human inputs and rigorous metrics that'll help say, wow, the big trial is probably worth doing. It helps validate animal models. I showed one example of that. There's lots more, and permits unbiased discovery opportunities that exploit natural human variability. And we've been part of lots of genomics and proteomics and metabolomics efforts, and I'll spare you that. It doesn't present well, but it is another way of discovering targets for therapy. And models like that microtissue model we view as one of the tools to study those hits that we get from unbiased screens. And I'll spare you the details on that. So if we come back to this, I am also interested as a way of introducing in being part of the translation to patients, especially phase 2 clinical trials, occasionally proof of concept phase 1 trials. In the Heart Failure Network, like the group here, we've been part of the series of studies that's been performed there. The one that I had the honor of leading was the FIGHT trial. This was within the Heart Failure Network. It was a phase 2 randomized trial of liraglutide and placebo-treated patients. Liraglutide versus placebo in high-risk patients with heart failure. By high risk, it was class 3 patients by and large with HF-ref, with hospitalization that got them into the trial, and most of them had a hospitalization earlier in the year. They were on significant dose of diuretics, so these were 3 to 3B patients. They were pretty sick. We included about 68% that were diabetic and the rest non-diabetic. We were hoping for a beneficial effect of liraglutide. As this slide shows, it was not beneficial. If anything, in the diabetic subgroup, which represented about 2/3 of the patients, there was a very, very close to harmful effect, right? This was not powered to show a difference on death or heart failure hospitalization, right? This is a 300-patient study, and there were only 198 that had diabetes, and yet we almost got significance for harm. And the curves are diverging, and the only time they're not diverging is when we're doing the run-in. So this was a very concerning slide, and it shows you, at the end of the day, you have to test your hypotheses. We had reason to believe that liraglutide would be a good thing, and in fact, GLP-1 antagonists or agonists are very favorable in earlier phases of heart failure, but in this later, it doesn't appear to be the case. I'm also involved in some proof of concept studies. One of the ones I'm most excited about is one we just got started in the past couple weeks, the EMAP study. This is a study where we all know that native heart biopsies are infrequently done in people with non-ischemic cardiomyopathy, even though we don't really understand their disease most of the time. We wondered whether we get a better yield if we use electroanatomic guidance, and we've begun doing that at Penn and at Mayo, where they actually led the way. And so we're doing a paired study design. Right, so this is the kind of voltage maps that our EP colleagues are generating all the time. They show us areas of scar, intermediate voltages, and normal myocardium shown in purple here, that we can get this with voltage mapping. And of course, our EP colleagues use this to decide what areas to ablate and to help disrupt arrhythmia circuits. But you can also get unipolar maps that can get more full thickness indications of where scar is and where intermediate zones are. There was a nice publication out of Mayo a few years ago that showed that there's characteristic voltage and electrogram properties of areas that are normal myocardium, let's say greater than 5 millivolts, are areas that are normal. Areas that are very low amplitude are likely scar, very low amplitude here. And that if we have intermediate voltage with a fragmented electrocardiogram, they're the more interesting places. And when they applied that in a case series, no controls, they found fairly high yields of interesting diagnostic findings, whether it was sarcoid or giant cell myocarditis, when they biopsied in the areas where there were fragmented electrograms and intermediate voltage. In areas where there was sort of a monophasic, very low voltage, usually scar. In areas where it was greater than 5 millivolts, usually normal myocardium. And so this needs to be tested about whether this is a way to get a better yield. And we got the company that makes the mapping system, Biosense Webster, to fund us to do this study. So a goal, enhanced diagnostic yield of heart biopsies. We'll do routine biopsy first under fluoroscopic guidance, targeted biopsy guided by electrical mapping. Wherever the mapping takes us, we have IRB approval to do it in the RV, the LV. It's under guidance from intracardiac ultrasound. We'll do established tissue analyses on both, and we'll see if we have a better diagnostic yield. We'll do this on about 60 patients at Penn and the Mayo Clinic, and the paired design will allow us to explore whether this approach of more targeted biopsies will give us a better diagnostic yield. Again, this is part of a bigger, sort of, if you will, precision medicine paradigm where we're saying this is what we usually do. We have new onset cardiomyopathy. When we do biopsy, we usually get a diagnosis of myocarditis, maybe 10% of the time. That's what the IMAC studies showed. Occasionally we stumble upon something else, but most of the time we get non-specific findings. Myocyte hypertrophy with diffuse interstitial fibrosis kind of readout. And the question is, would we have a better, again, this isn't the answer, this is the hypothesis, that the non-specific findings will be lower if we sample better. If also we should be doing genetic testing on more of these patients. So it's part of a bigger precision medicine paradigm, but I think the better tissue sampling is part of it. Another part of it is that something like the electroanatomic mapping approach, it doesn't just guide the biopsy, it actually tells you extent of disease, right? So if you'll find out that area that ended up being inflammation or suspicious for inflammation, you'll find out whether it represented a really large or smaller part of the myocardium. And so we're being more like oncologists. We're biopsying more and more of the cases, we're staging the disease, and maybe as we use these better samples to study in a lab, we'll also get better insights into disease and more targeted therapies. That's a more forward-looking goal. For now, we want to see, can we get better diagnosis and maybe a little better prognosis from staging their disease? That will start to address in this trial. The targeted therapies are for the future. So I'll stop there. Again, the acknowledgement for this part were the FIGHT trial. Again, all my colleagues and many others not listed here in the Heart Failure Network. The EMAP trial with people at Penn and Mayo. Again, some funding from Biosense Webster for the EMAP trial and from the NIH. And I'll stop there and thank you all for your attention.
Thanks, that's great. So we're open up for questions, and again, use the microphones because we have some people listening remotely. I'll start with one question. So back to the first part of your talk where you're talking about the microtubules and their effect on cell stiffness. What about the other potential interactions of the microtubules? So in the vasculature, they've been linked to potential nuclear signaling, sort of transducing or transmitting the tension to the nucleus, or as sort of an escalator carrying mRNA, signaling molecules. What are your thoughts about potential other roles there?
So absolutely, we're interested in those questions, and there's collaborators at Penn who are exploring those, and particularly, especially the nuclear signaling aspects. And we're just beginning to tell you the truth, but I think those are relevant things. What we don't know is whether the tyrosination-detyrosination balance is something that's most important for the biomechanical effect, or it also has implications for the nuclear signaling. It might not, but that's really what we have to find out. They could be sensing, but only resisting contraction as one effect, but still have the same perfectly intact mechanosensing role, irrespective of tyrosination-detyrosination balance. So I think we'll surely find that something like colchicine that really disrupts them in toto will probably have an effect on nuclear signaling and other things you mentioned, trafficking of nucleotides. But whether tyrosination-detyrosination balance will modulate those other functions of microtubules is uncertain.
Some other questions?
Yeah.
Okay, just a really simple question. Does the buckling, what exactly is that indicating?
It looks like it's attachments to Z lines. Okay,
So as the buckles form, are they preventing or limiting the shortening that's occurring?
it certainly looks like that because, okay, so
And how could one experimentally say that actually is important?
okay, so again, there's a whole the manuscript from Prosser's group from 2016 kind of goes into quite a bit of detail of that, but suffice to say that the best way to do it is to shift that balance of tyrosination or detyrosination and then repeat the biomechanical assessments, which is what we've done in both the rat and the human cardiac myocytes using rigorous in vitro approaches. So I think the way you show that the buckling, again, it's by association, but that manipulations, pharmacologic or genetic, that affect the tyrosination-detyrosination balance, that affect the mechanical buckling characteristics, and this is the part I probably didn't present enough of, is that the characteristic wavelength gets progressively longer as you tyrosinate, because there's fewer attachments to the Z line. And so you have fewer bigger buckles that have less resistive effect. And when you have the more places that you're actually anchored to the Z line, the more integrity or the more resilience this microtubule network has.
Is occurring exactly in the same position.
It's well, depending on the tyrosination-detyrosination balance, yes.
No, I mean that when you're looking at the video images, and you're saying that these...
Oh, the buckles are, yeah, they're reproducible.
It's not occurring randomly.
No,
it's occurring
no,
at those
no.
points.
Correct.
I see.
And even the second video where we slowed it down, you can see it happening again and again. But yes, so it happens at the same points with every beat in a given state. But then when you manipulate the tyrosination-detyrosination balance, then you get a generally longer average wavelength. So it starts, instead of spanning one Z line, one sarcomere length, it starts being 2 or sometimes 3. And that those buckles don't appear to be as stiff. What you don't get is them just sliding next to each other. There's always some residual anchoring to the Z line. So there's always some buckling. It's a matter of what's the periodicity of that buckling.
Any more questions from the group? I think given the time, we'll go ahead and thank you very much for a good seminar. Nice having you here.
The preceding program is copyrighted by Emory University.